And again, I'll be here if you ever need anything.
Okay, perfect, Ali. Thank you. Okay, Dr. Plack, so I think we can start with our series. First of all, welcome to our Aero International lecture series. This series will highlight the speakers from around the world and give them a platform to discuss their career and their research. Today, our first speaker will be Dr. Chris Plack from the United Kingdom. Dr. Plack, thank you for accepting our invitation.
Thank you. Thank you for inviting me.
Of course. So whenever you want, you can start. So please feel free to leave your questions in our chat, and these will be answered at the end of the session. Thank you.
Great. Thanks very much, Melissa. So I was thinking about a title for this talk, and I thought my noisy life kind of covered some of my research interests and some of my recreational pursuits as well. And it's also maybe a sort of metaphor for some of the things that happened during my career in terms of directions I've taken. I've been a bit stochastic. So that's the title. And following the instructions, just see if I can get this to work. Okay. So first of all, I'm going to talk a bit about what's described as in the guide as my research journey. So I'm going to tell you a little bit about my career to date, what I've done, who I've worked with. I'm going to tell you about some of my current research interests. And then finally, I've been asked to talk about some of the challenges faced by hearing researchers in the UK. And then finally, I think we get a question and answer session at the end. Okay. So there are many ways, I suppose, of me describing my research journey. So the first one is just in terms of the places that I've worked during my career. So I started off my undergraduate degree in natural sciences at the University of Cambridge, and then I did a PhD in the Department of Experimental Psychology. I then had a 2-year postdoc at the University of Minnesota. And then to escape my creditors and the Inland Revenue, I moved back to the UK, University of Sussex, also a postdoc, and there obtained a Royal Society University Research Fellowship that helped a little bit make me an independent researcher. I then took up my first, I suppose what you call tenured position at the University of Essex as a senior lecturer, was promoted to professor chair in 2001. Then in 2005, I moved to Lancaster University, and then finally in 2008, I moved to the University of Manchester. But in 2013, I kind of split myself and half of me went back to Lancaster University and half of me stayed at Manchester. So I currently have 2 appointments, one at Lancaster University and one at Manchester. They're only about 50 miles away on the M6, which is a motorway in the UK. So it's quite easy to get between the two. So I have 2 jobs at the moment, one in the Manchester Centre for Audiology and Deafness at the University of Manchester, and one in the Department of Psychology at Lancaster University. So another way to look at my research journey, and probably the most significant in terms of the influences, to look at the people that I have worked for and with. So the University of Cambridge, I was very lucky to be supervised by Professor Brian Moore. I'm sorry, this is jumping a little bit. I'll try and get the sequence right. And Brian has really been an incredible influence right from the start, really. I started off getting sort of interested in hearing science by doing some experiments as a participant in his laboratory. And then I was lucky enough to get a PhD position with Brian, and he's really been a guiding influence throughout my career. So most of what I've achieved, I think, is probably thanks to him. I then took the postdoc at the University of Minnesota, where I worked with Neil Viemeister, who some of you may know is sadly recently passed away, but he was also a sort of formative influence, gave me a kind of little bit of a different perspective on psychoacoustics. So this isn't, there we go. This is not progressing very well in terms of the animation, but anyway. So yeah, so I worked with Neil for just 2 years, but again, we kept in close contact throughout my subsequent career. I then moved to the University of Sussex and took up a postdoc with Bob Carlyle, and I think these 3 influences really sort of cemented my knowledge and my outlook really on the whole field of hearing science. And I think it's very difficult to pick 3 people you'd rather have to be your teachers and mentors, I think. Around this time as well, I started up a collaboration with Andrew Oxenham, now at the University of Minnesota, of course. And we started, it was really based on Andrew's PhD thesis, where we were looking into ways of measuring basilar membrane nonlinearity using behavioral techniques. And this was also a really important experience in terms of learning how we can combine psychophysical methods and learn about the underlying physiology. When I moved to the University of Essex, I did some work with Ray Meddis. And Ray, as some may know, was rather, again, sadly passed away, one of the foremost computer modelers mainly of the peripheral auditory system. And I learned a lot from him about some of the approaches to computer modeling. And that, again, was a really useful experience for me in developing my technical ability, I suppose. When moving to Lancaster, I started well, slightly before I moved to Lancaster, but during this period, I developed a collaboration with Deb Hall, and Deb, as many will know, is an expert on auditory functional magnetic resonance imaging. And so that was another interesting technology, although I can't profess at all to be an expert on MRI. It was, collaborating with her gave me the opportunity to do some sort of brain imaging, which helped sort of develop some of my understanding and some of my knowledge of pitch perception, and helped to sort of look at new ways in which to investigate the representation of pitch in the brain. And in a similar vein, Ravi Krishnan, he came over to Lancaster University for a sabbatical, and he taught me the basics of the frequency following response technique to measure phase locking in the brainstem. using electrophysiology, and this started my interest in auditory electrophysiology, which has persisted to the current day. Okay. So finally, University of Manchester, I still work quite closely with Professor Kevin Munro, and Kevin is one of the, he started off as a clinician, but he's moved and become a very successful researcher. So through him, I really got a lot of insights into how to translate some of the basic science findings in hearing research into clinical applications. So I owe him a great debt in terms of opening my eyes to some of the sort of translational opportunities in my work. Okay, so those are the places, those are the people, and then we can talk about, I mentioned it a little bit, but some of the techniques. So I started off pretty much as a hardcore psychophysicist, as you might expect from having Brian, Neil, and Bob as my supervisors. And this continued until, I mean, I did a little bit of modeling for my PhD, and I've always been a little bit interested in computational modeling, but this really kind of took off when I worked with Ray, who taught me a lot about how to develop models of the auditory system and how to test them against the data. And then, as I mentioned, my work with Deb Hall got me interested in auditory fMRI, and we still do some collaborations with the group at Nottingham. And with Ravi, having a look into ways of using electrophysiology to sort of try and bridge the gap between the sort of behavioral measures that we use and the underlying function of the auditory nervous system. And then while I've been at Manchester, I've developed interests in using more sort of clinical tests, sort of obviously audiometric techniques, otoacoustic emissions, and middle ear muscle reflex, and also using survey techniques. This is more recent, and some of this was forced on us by the pandemic, using lots of online testing, but we're doing quite a lot of that now. We've developed sort of ways of getting sort of subjective responses about people's hearing ability. And then finally, the sort of most recent, I suppose, technological development in my career has been the using this assay, the ELISA assay, which is a way of measuring protein concentrations in the blood, and we've been using it to, there's a certain interest at the moment in seeing whether Prestin, which is the molecule expressed in outer hair cells, is responsible for electromotility, whether we can actually measure that in the bloodstream and perhaps use it as a biomarker for different types of hearing disorders. And then finally, in terms of my journey, I can talk about the sort of things, you know, the actual topics that I've been interested in. So I started off in my PhD looking at temporal resolution and the temporal window model of temporal resolution. With Neil, I did some work on intensity coding. With Bob, I got interested in pitch perception. See if this falls, yeah, there we go. Sorry, it's all a little bit jerky, but there we go. And then, as I mentioned, with Andrew, we started to do some work on cochlear nonlinearity and using behavioral techniques to measure the response function of the basilar membrane in humans. I then, with the fMRI and electrophysiological techniques, and also working with Bob and Hedvig Gokul, doing a lot of work on pitch perception and looking at, again, using behavioral and electrophysiological techniques to explore pitch coding in the brain. And more recently, we've been doing a lot of work on the effects of noise exposure on hearing, particularly the sort of subclinical effects and damage to the auditory nerve. And so noise exposure has become quite a big part of what we're interested in, particularly at the University of Manchester. Also interested in the effects of aging and in comorbidities, that's sort of diseases such as diabetes and dementia that often co-occur with hearing loss, and trying to understand the relationship between them. So that's my kind of diagram of my research journey. I hope that kind of makes some sort of sense. So it's lots of different things going on here, but it's kind of interesting actually just thinking back at this, sort of thinking about the progression of how, started off, with based around sort of behavioral techniques, and how as I've gone through the career I've sort of added different things. And I think this has been really useful, having sort of different ways of sort of exploring the same kinds of questions. So my current research interests, this is sort of one way of expressing them. I found it hard to sort of conceptualize really, because I tend to sort of jump around and do lots of different things. But we're doing a lot of work on the effects of noise exposure, as I mentioned, also very interested in the effects of aging on hearing. And these are, these areas are really focused on the kind of subclinical deficits, and not the sort of conventional things that you see in say, pure tone audiometry, but looking more at the changes to the auditory nervous system in particular that might occur through noise exposure and aging. We're also interested in the effect of diabetes on hearing. We've done some work recently on the effects of COVID. We are continuing to do a project on the effects of COVID-19 on hearing. There's some indication that it might have, obviously we know about the effects on taste and smell, which have been widely reported, but there's some indication that COVID-19 might have some effects on the auditory system. And so we've been doing various things to try and explore that. And we're also interested, we've got a project looking at the effects of radiotherapy treatments to head and neck cancers on hearing. And then another, I've sort of grouped these together, but they're not necessarily strongly related, looking at the effects of the relationship between hearing cognition, the relationship between hearing and dementia in particular, trying to explore the underlying causes of these relationships. And also through my PhD student, Kate Blackthorne, we've been having a look at the auditory abilities of autistic people, which is a very, very interesting area, and it hasn't really been explored from the perspective of autistic people. So we tried to get some input from the autistic community to guide our research. And Hannah Guest, who's a postdoc at Manchester, has also been instrumental in putting this together. And so we have these different areas, and as I mentioned throughout my career, I've sort of tried to sort of pull together all these different types of techniques. So we're using a range of different methodologies, from psychoacoustics, extended high frequency audiometry is something that we've been exploring a lot recently, electrophysiology, fMRI, and then surveys, and then finally these Preston assays using the ELISA technique. So I just thought at this point I give you a kind of just a sort of smorgasbord of some of my recent research findings, just to give you sort of flavor of some of the things that I've been up to in the last sort of 8 years or so. So I mentioned that noise exposure is a big interest of mine. Here is it's actually a null finding, so it might have been that exciting to people, but we've been going after cochlear synaptopathy in quite a big way over the last few years. And this is one of our early findings that was negative from Gareth Prendergast, was a postdoc on this project. And this is showing auditory brainstem responses, so the electrophysiological responses, and the dashed lines here, the red is female, the sort of turquoise color is male participants, and the dashed lines are for participants who self-reported low noise exposure in their lifetime. These are all young people with clinically normal hearing. And so the dashed lines are for low noise exposure, and the continuous lines are high noise exposure. And we were looking to see whether we could sort of spot changes, particularly in wave 1 of the auditory brainstem response, which reflects auditory nerve activity. And as you can see from this, we didn't find very much evidence, at least in our sample. that there was very much change in the auditory brainstem response. So we didn't find much evidence from this of cochlear synaptopathy. In Hannah's PhD, Hannah Guest's PhD work, she amongst other things, she had a look at the effects of noise exposure on tinnitus and also the relationship between tinnitus and neural deficits. And in this particular study, although we didn't find a link between tinnitus and neural loss, at least as measured using the auditory brainstem response, we did find that tinnitus, these people who have totally clinically normal hearing, and we actually match their hearing up to 14 kilohertz. So there's very little auditory the difference in the groups in terms of their basic audiometry, their basic sensitivity to sounds. But some people with normal audiometry do report tinnitus, and we found this seems to be quite strongly related to their noise exposure history. So that gives a little bit of confidence that our noise exposure measures weren't just entirely random, that they were telling us something about the experience of the individuals, and that these individuals did seem to have a link. So noise exposure, even if you've got completely clinically normal hearing, does seem to be related to tinnitus. So let's see whether this is going to come up. Sorry about this. Okay. So we have had recently, this is some data that's hot off the press. It hasn't been published yet, from Adnan Shahabi. He's a PhD student of mine at the moment. When the pandemic happened, obviously we had to sort of switch research methodologies in the UK, at least. I'm not sure whether this has been, how much this has been the case in other countries, but we pretty much had to stop all in-person testing for a year or so. So we moved things online, and this was kind of an interesting experience in developing some of these online protocols. But we developed an online speech in noise, a suite of speech and noise tests that we could use online. And this is some data that we collected using that. Now, these are workers in Palestine. And in Palestine, the reason Adnan was interested in this, because his community in Palestine, he knew that there were a lot of people who were exposed to very high noise levels from working in noisy factories, and there didn't seem to be as much control or regulation of noise exposure. And so this is an investigation of that. These are people who all self-reported normal hearing. We couldn't measure their audiograms, this was online, but they self-reported normal hearing, and they didn't have any diagnosis of hearing loss. And what we found in these recent data is that there was quite a strong relationship between the occupational noise exposure and performance on this digit in noise, speech and noise test, so that people with more noise exposure seem to perform quite poorly. And you can see that for most noise exposure here, the performance is really a lot worse than it would be for a healthy ear. And then finally, I just wanted to sort of pop this up. It's often not recognized, is that, you know, we think about occupational noise exposure as being noisy workplaces, noisy factories, but musicians who have professional musicians, they're often exposing themselves to very, very high noise levels. And this is just a little indication of this using online methodologies to measure noise exposure. It just sort of shows that musicians working in orchestra, so it's not necessarily rock musicians, also classical musicians, people working in the music industry in general, they tend to expose themselves to high levels of occupational noise, often without hearing protection. So those are kind of some of the things we're doing on noise exposure. I just want to expand it a little bit, having a look at, I mentioned subclinical deficits. These are deficits that don't show up in normal clinical testing. And these are some of the things that we're trying to probe, particularly using electrophysiological techniques. So this is some work that former PhD student Ollie Bones and I did, looking at the representation of musical stimuli in the brainstem using the frequency following response. So this is two bursts of a dyad, so a two-note chord with an interval of perfect fifth. And this is showing the impact of age on neural coding in the brainstem, which is can be really quite dramatic. So these two groups are only, the difference in the groups, the cutoff between the groups was only 40 years old. But you can see there's quite a difference in terms of the phase-locked response. The young listeners, shown here in the blue, show a much stronger phase locking to these musical stimuli than do the older participants. By older, it's only 40 years, so they're not really that old, but it does seem that you get these quite dramatic changes in terms of the neural response to these stimuli, and we have other evidence to suggest that that is related to the perceptions of these stimuli. So as you get older, you tend to show less differentiation between different musical intervals. So perception of harmony is dulled a little bit. The next bit of data I'm showing you here, this is Awa Al-Jasser, a former PhD student, looking at the effects of diabetes on hearing, again using the frequency following response technique. And these are type 1 diabetics, and they have, again, so they're all young, clinically normal hearing right the way up to high frequencies. I think we went up to sort of 12 kilohertz or something like that. So the groups were well matched in terms of their audiometric sensitivity, but as you can see, the phase locking to both the envelope, that's the overall fluctuations of the sound, and the temporal fine structure, the individual pressure variations, they're reduced for the diabetic group. And so that suggests that diabetes is having some effects on the neural coding of stimuli that isn't that isn't reflected in any kind of loss in audiometric sensitivity. The next one is an investigation of the effects of aging using the auditory brainstem response, and this isn't, we're not the first people to discover this, but this is just an illustration of the reduction in the response, particularly in wave 1 of the auditory brainstem response, as you go from a group of young participants to more elderly participants, sort of 60 plus in this group, and you can see quite a dramatic reduction in the amplitude of wave 1, reflecting the auditory nerve response. And this might reflect cochlear synaptopathy. This wouldn't be that surprising if it were just in isolation, because if you lose, although the groups were matched pretty well in terms of their hearing up to about 4 kilohertz, at high frequencies, they weren't that well matched. But even after we controlled for the high frequency loss, statistically, there was still quite a large difference between the groups in terms of this measure. So this might be indication that there's something, that there's some loss of auditory nerve fibers or cochlear synaptopathy or something like that. And finally, I just wanted to show you, we're sort of getting, as I mentioned, pretty interested in extended high frequency audiometry. That's testing frequencies above the range that are normally tested in the clinic. And this just shows you, so we have two listeners here, the green and the purple, who have normal hearing. In fact, their hearing in this particular illustration is matched, almost identical up to 8 kilohertz, but they vary massively in terms of their hearing thresholds at higher frequencies. So 16 kilohertz is about sort of almost 50 dB difference between the two. And so this is kind of an unexplored, well, it's not totally unexplored, but it's an area which we think has a great potential as for giving us the early warning of hearing damage, particularly things like noise exposure. It's well known with ototoxic medications, you tend to get losses in this extended high frequency region as well. So that's a useful future target for audiometry, we think. Okay, so I'm just going to finish off this bit. Sorry, how am I doing for time, by the way? Sorry, I've lost my clock.
We are okay with time, Dr. Plack.
We're
have
okay.
enough time, so don't worry.
Sorry,
Go ahead.
Fine. Okay. So I just thought I'd sort of throw in some of the, as I said, my mind tends to sort of go off in some strange directions. I meet some interesting people and I start some interesting collaborations, and I end up doing some things that I wouldn't normally sort of consider. And so this is an interesting one we did. This is a research fellow Jenna Littlejohn at the University of Manchester, and we did some work during the COVID pandemic, looking at the impact of the pandemic for people with hearing loss. Because if you've got an auditory disability, then it could be that you're less able to communicate easily with your friends and family if you're in isolation or shielding. And we hypothesized that this might have an impact on your mental health, and possibly also in terms of your cognitive ability. There's a possibility that the sort of interactions that you have in your social life that are reduced during the pandemic might have some sort of impact on cognition. And so this was a self-report survey. These are all people over the age of 70. And this particular set of plots shows whether they reported a change, a worsening, or no change in terms of these different measures. So we did a clinical measure of anxiety, a clinical measure of depression, and we also had a measure of memory. And what we found, basically, first of all, that people with hearing loss reported. So this is the SSQ-12, which is a self-report measure of listening ability. And so higher numbers here represent better self-report listening ability. And so we found that people with worse self-report listening ability tended to show a greater increase in anxiety, an increase in depression, and also a worsening in their perceived memory. Of course, you have to be a little bit careful here because this is all self-report. So this is an indication that people with a hearing loss are particularly vulnerable to the effects of enforced isolation that occurred during the pandemic. This is another little sideline. I mean, one of the reasons I got into hearing actually is because I play guitar and I played in bands and I was interested in music, and so I kind of got into being interested in what goes on in the ears when you listen to music. But this particular thing looks at it from a slightly different angle, and so I sort of hooked up with this guitar maker in Penrith called Roger Bucknell, of Filed Guitars. He's a really great bloke, and he actually was originally trained as an engineer, but he hand makes these really beautiful steel-strung acoustic guitars. And he collects wood from all over the world and tries out all these different woods. And as some of you guitar players will know, there's quite a lot of premium placed on guitars that are made for particular sorts of woods. And so what we did is we got him to make 6 different instruments where they're identical in all respects, apart from the woods used from the back and sides of the guitars. And then we got professional musicians and semi-professional musicians and amateurs to play them in blinded conditions. So they're in a darkened room wearing welder's goggles, so they couldn't really see what the wood was. And then to rate the guitars. And this research was conducted by Sam Carcagni, who's a former postdoc of mine at Lancaster and a former PhD student. And what we found basically is that the back and sides of the wood of the guitar had little effect on the overall perceived quality of the guitar and the playability. And we also did ABX testing, comparing instruments with different woods to see if they could tell the difference, and the D primes were all fairly low for that. So this is kind of had a little bit of a sustainability angle, really, because some of these woods, like Brazilian rosewood in particular and Indian rosewood, the rosewoods are actually limited now in terms of the import and export because they're endangered. So it's really kind of having a look and seeing, do we need these really expensive tropical hardwoods in order to make guitars? And this study suggests that you don't necessarily need those. Any differences might be sort of quite subtle, at least for the back and sides of the guitars. It probably doesn't make that much of a difference. And then finally, I just thought, because I mentioned the Preston work we've started doing, which we're quite excited about. It's a bit of a leap of faith to see whether, imagine that you have 12,000 odd outer hair cells in each inner ear, and to see, you can imagine how, to compare the trillions of cells in your body, the small amount, the tiny amount of prestin, this protein in the outer hair cell membrane that might be released if the hair cells are damaged. But you can detect that, and there's been some interesting animal work suggesting that you can detect these minute traces of prestin if there are damage to the outer hair cells. And so we actually started doing some, I don't have any of the recent data, which might be, we're still sort of collecting the data, might be quite exciting in terms of the effects of noise exposure. But Eleftheria Iliadou and some other colleagues at the University of Athens have been collaborating with me on this. And these are some of Eleftheria's earliest results, just having a look, and so it's not really an auditory thing, but having a look at how prestin concentrations vary according to the circadian cycle, just to see, is one of the sort of baseline measures that we wanted to do. And so this shows you can measure the prestin levels in blood serum down to 20 picograms per milliliter. So you can detect these sort of tiny trace amounts of prestin in the blood. And so potentially this might be a sort of useful biomarker for detecting damage to the cochlea. Okay. So that's just a kind of an overview of some of the recent findings, which gives you a little flavor, I hope, of some of the things that I've been doing for the last few years. And so I'll just finish off by, I've been asked to talk about some of the challenges for hearing research in the UK. And some of these are going to be common across many of you, I suppose, but I might as well mention them anyway. So the first thing is lack of time, is just having time to do research. And there are, I suppose, as you get, as you progress in your career, there are more sort of things that, in terms of the academic life, that kind of interfere with your time that you have for research. But given that, and given the fact that obviously I'm expected to take more of a role in administration and management. I think, and I'm sure many people would agree, that the admin load at universities, particularly in the UK, seems to have increased dramatically over my career, and a lot of this is down to meetings. Just look in your diary, and you just see that you have a week, and basically you have no time to yourself. You've just got meeting, then another meeting, then another meeting. And so I think this sort of sums it up. Meetings are a practical alternative to work. And it seems to me that often, that universities are just one of the mechanisms for creating more committees, So that's one of the things that occurred to me that has changed in my life. As I say, it's partly the fact that as I become more senior, I have to attend more of these things, but it certainly interferes with the time I have for research. And then there's bureaucracy. There's just so much paperwork now. I mean, some of it's good, of course, in terms of ethics and risk assessments and things like that, and ensuring sort of safety. But there still seems to be an enormous amount of bureaucracy in everything you do, and this kind of tends to slow everything down as well. The other thing, of course, is lack of money. We're all going to complain about this, but there's been a sort of bit of a squeeze in terms of government finances. Obviously, we've had the pandemic, although there is a sort of move to push more money into research. I think that there has been a certain squeeze to the research councils. And then we're still waiting to see what's happening with the fact that the UK has left the European Union. We used to get a lot of money. Well, we still do get a lot of money through the EU and for the European Commission. And we don't know what's going to happen with that. So that's a little bit of a worry. And then the thing is, if there's little money around and you have sort of success rates on grants of 10% or something like that, this actually impacts on the time as well, because you spend more time applying for money because it's harder to get. So you spend more time writing grants. So this is something that I found, maybe I'm just getting worse at writing grants, I don't know, that I seem to spend much more time writing grants and perhaps reviewing other people's grants than I do actually doing research. So that has that impact. Another thing that I think is a kind of general problem maybe with hearing science is that there's a kind of lack of recognition of the scale of the problem. You know, it's one of the most significant, I think it's the World Health Organization has this as the third leading cause of long-term disability worldwide, and we know it affects 1.5 billion people worldwide, and it's estimated to cost the world economy about $1 trillion a year. So it's quite a big deal. But in the UK, at least, I don't know what it's like for other countries, only about 0.6%, 0.6 of 1% of health-related research funding goes on hearing science. So I think there's a real gap between the actual scale of the problem and the amount of money that's being put in. So there's a kind of lack of recognition there, and that sort of feeds back into this, into the lack of money. Perhaps if it was a priority area, then maybe there'd be more funding. There's some other things that occurred to me just sort of thinking about the sort of limitations. One is working with external organizations I find quite challenging. I don't know what the experience of other people in the UK is, particularly working with the National Health Service, sort of again, sort of bureaucracy and admin and sort of getting permissions in place and things like that, and just find, clinicians are very, very busy in recent years, of course. Even more so, but it's actually quite hard to, I've found it quite hard to sort of find those links. And also with industry, it's getting these sort of communication channels and these sort of structures in place that enable you to collaborate easily with these external organizations, which I think would be really, really useful, which I find, personally, I find this is sort of a sort of barrier. I wanted to mention as well, this isn't just a UK related thing, but these sort of science practices, because I think that hearing science is a little bit slow to sort of catch up with this sort of movement that's happening in neuroscience and in psychology, to do with what's being called the sort of replication crisis, where you have the preponderance of false positive results in the literature, where research findings are published and they look fine, and then maybe they haven't been able to replicate, and this can be quite damaging because it can obviously lead researchers down wild goose chases, and the literature sort of fills up with findings that don't actually advance the field, and they actually sort of send you in the wrong direction. And so there's this was really highlighted in psychology, where a lot of sort of seminal results they found that they couldn't replicate. And I think we have to sort of take a little bit of a look at ourselves, and I sort of count myself as one of the villains here in terms of the practices. In the early parts of my career, it's sort of the idea that you're always searching out for the positive finding rather than trying to publish all your findings however they come out. And so one of the sort of key things is to have a sort of pre-registration or some way of documenting what you originally set out to do and how you originally set out to analyze your data before you collect the data. So there's always a tendency to collect the data, then go fishing through it, and then trying to find out those results that are that sort of pop out, that you can then get your nice splashy publication. So I think we're sort of wising up to that a little bit, and the importance of transparency about what you actually intended to do and how you intended to analyze your data. And also this sort of public availability of data as well, I think is very important. And meta-analyses in particular are sort of great ways to actually discover to sort of get the underlying truth from a number of different studies. And if we all make our data publicly available, these become much easier to do. But also, if the data are publicly available, people can sort of check through, make sure you've, that your methods are sound in terms of your analysis techniques and things like that. So I think making data publicly available and pre-registration are two of the things I'd like to see more of in our field. And I think that we're a little bit behind the curve in terms of what's been going on in neuroscience, perhaps. And then finally, I just thought I'd mention, one of the sort of challenges is increasing the diversity of what we do in terms of getting different populations, both involved in being scientists and and doing research and having different perspectives. It's a sort of standard thing that a lot of us are white middle-class males. And having some sort of diversity, I think, will be really helpful. So there's that, and getting these different perspectives, and of course equal opportunities. But there's also, I think, a lack of diversity in terms of the actual populations we study, which tend to be university students, let's face it, for a lot of our studies. And we tend not to go to more in sort of impoverished backgrounds, or to study, like Adnan's done with his study in Palestine, study different sort of populations who might have some different sort of issues that are worth considering. Is also getting the sort of patient and public involvement as well. So you hear from these different communities and learn about what they want us to do research on, rather than what we think we should do research on. Okay, I think that's just about me for this part of the presentation. I guess I just have to finish off by thanking if I can get to the final slide. Is it coming up? Ooh, very slowly. Oh, it's just ended. Okay, sorry, there we go. Some of the funders who sponsor the Medical Research Council, Biotechnology and Biological Sciences Research Council, National Institute for Health Research, who funds the Manchester Biomedical Research Centre, which has been a great source of funding for my research at Manchester, and also the Royal National Institute for Deaf People, which is the main UK charity for people with hearing impairment. So thank you very much. And I think we're now over to the Q&A session. So I don't know whether this is going to happen in the chat or whether it's going to be live.
Thank you, Dr. Plack, for this wonderful presentation. We are really happy with this first lecture series. So we have some questions in our chat. Let's start with the first one. It's from John Allen. Is there any evidence that COVID attacks cilia of inner ear or outer cells?
It attacks the, which, sorry, I'll just see if I can see it. It attacks the stereocilia, or the, oh, okay. Oh, the cilia, okay, sorry, yes. I don't know, that's a good question. I don't know the answer. I mean, obviously, if there's inflammation, you can get all kinds of things going wrong. There might be an effect on the blood supply. We've been thinking of it from a more sort of neurological perspective, that it might be affecting the auditory nervous system. But I don't know. So the evidence so far is largely one of the problems that there haven't been any really good prospective studies so far, so that most of it's come from people self-reporting deficits. But we did do a study with Al Jassa in Saudi Arabia, my former PhD student, which is a self-report study, which when we had an actual control group of non-COVID patients, they also reported a decline in their auditory abilities. So we didn't find any significant effect of COVID. But yeah, our thinking at the moment, I'm not sure about the cilia. Our thinking at the moment was more to do with inflammation affecting the auditory nervous system. And this is one of the mechanisms that we think is leading to people having difficulties with smell and taste. But yeah, I don't know. I don't understand enough about the mechanisms of inflammation and other types of things that go on when you get infected to give some sort of expert opinion on that.
Well, we have another question. Your results on the relation between noise exposure and speaking noise performance seem to show a non-linear relationship. Would you agree? What sort of exposure is needed before you see a distinct worsening of speech and noise perception?
Yeah, thanks, Brian. Yeah, that's a good point. It does definitely seem to be some sort of curvilinear relationship. I'll have to go back and have a look and see what the actual level of exposure was. I mean, that scale was a logarithmic scale. So in terms of the total energy of exposure. So I think we'd have to sort of go back and have a look and see whether the sort of exposures that people are getting in these factories, what sort of I think we do know enough we can relate it, I think, we should be able to relate it to the actual exposure in terms of dBA, LEQ or something like that, that these people are having. So we should be able to do that. But yeah, but that's a very good point. I mean, we've just drawn a straight line through it, but yeah, it definitely seems to be, nothing much happening, and then it kind of shoots off and you start to see these big effects on speech perception. So yeah, that's an interesting point. So we'll have to explore that. I think
well, besides other cells, Preston is also expressed in heart. How do you separate heart problem from hearing issues?
Sorry, could you say that again, Melissa? How do I separate?
Sure. Besides other hair cells, Preston is also expressed in heart. How do you separate heart problem from hearing issues?
No, that's a great question. I guess that it depends on the population, right? So if you've got some of the experiments that Eleftheria is doing is on young people who we hope not going to have heart problems. So obviously, you can sort of screen, in a given study, you can screen out for people who've had cardiovascular problems that might be involved in this. So one of the things we're interested in is, I'll just tell you a little bit more about what we're doing. We're actually looking at sort of short-term effects, because some of our data so far suggests that maybe you're not going to get long-term changes in Preston in the blood. You might get long-term depression. But what seems to be happening, it seems to be the case in the animal literature, is you get a short-term uptick in Preston concentration in the blood over maybe a couple of days or so, if you've had some kind of trauma, which kind of makes sense. If you damage outer hair cells, Preston's released. So I guess one of the potential ways in which Preston might be useful is giving you some idea about whether you've had some recent trauma. And so that it could be used potentially, and this is something we're going to have a look at as well in our studies of the effects of radiotherapy on the auditory system, and we're hoping to do a Preston assay for that as well, to have a look to see whether it could be a useful biomarker that you're actually, a sensitive biomarker that you're actually damaging the hair cells in the inner ear. So for that, so I guess for heart conditions, I'm not exactly sure the relationship between, I don't know enough about the literature to know the relationship between having a heart condition and blood Preston concentrations, or whether if you actually have a heart attack or something like that, whether that actually causes an uptick in Preston, I don't know. But if we're doing it, if sort of looking for these sort of short-term changes rather than that's the way I kind of see it at the moment. So rather than being something that might be useful for saying diagnosing hearing loss, so you just have a blood sample, great, wouldn't it? Take a blood sample and tell us whether you've got hearing loss or not. It might be useful as a biomarker that you might have had some short-term damage to the ear, and obviously, that might be something potentially that you could then use to maybe change the treatment or something like that, so that you didn't cause that you sort of stopped that progression of damage. But yeah, it's a good question. It's interesting that a lot of people assume that Preston is just in the inner ear, but actually, yeah, you're right, it is expressed in other tissues as well. Okay,
we have two more questions, Dr. Plack. One is, your Dieter-Wood study is consistent and their Ganger hardwoods.
Sorry, you froze halfway through that for me. I don't know if we got a bad connection or what. So, okay.
Hi, Dr. Plack, could you hear me?
Yeah, so sorry, you froze halfway through. Sorry.
Yes, I think it's my internet. It's my internet right here.
It's either yours or mine, I don't know.
We don't know. So this question is, your Dieter-Wood study is consistent with the large number of high and luthiers who have abandoned endangered hardwoods.
So, can I just read? I didn't hear that quite right. Is this a question in the chat? Is it?
It's from Jay Rubinstein.
Okay, guitar which is consistent, yeah, with a large number of who've abandoned. That's right. So it's kind of a comment, really. Yeah, the thing is that I think that's true, but there is still, and I know this from personal experience of the people who get angry when you try and do these things. I mean, Claudia Fritz, I know you're familiar with her work, did a similar thing. And this got inspiration for our work on the violins, where she kind of effectively showed that all these really expensive Stradivarius violins and things like that were actually, well, in her case, a little bit worse than modern violins and high-end modern violins, despite costing an awful lot more. And so there's I think it's going to be very hard to persuade people because there's this kind of a mythology and a certain amount of suggestibility as well, I suppose, in terms of people, because it's expensive, because it's beautiful, people think it sounds better. And I guess to some, from one point of view, it does sound better, even though objectively there might not be any difference in the acoustic waveform produced by these instruments. But yes, so I think some people are coming on, some luthiers are coming on board, and they're actually often using recyclable materials. I know Martin, sort of, some other companies have started making guitars with recyclable materials, but there's still this perception, and they still make these guitars with these really, really expensive. I mean, I don't know what a Brazilian rosewood Martin would cost. I mean, we're talking about tens of thousands of dollars, I think. So there's still a sort of perception in the community, and it's almost, I think, a little bit like sort of this sort of hi-fi community and these kinds of things that people think make their hi-fi sound better, and because they don't do any kind of rigorous experimental comparison between different sort of amplifiers or different connections between bits of hi-fi equipment, they convince themselves that they can hear the difference. So I think it might be quite different. There's a lot of people who still, certainly through some of the conversations that we've had, are still very resistant to the idea that you can test these things in the laboratory. But if you can't test them in the laboratory, then how do you test them? And so you get to this sort of impasse, really. So you can't do the experiment because people don't believe anything that we do in the laboratory, and yet we don't believe what they say, because that's sort of subjective. We know how suggestible people are. So but yeah, you're right, it is that people are sort of coming around to it. But I think the other thing is, part of the thing about these instruments is they are beautiful objects often, and so the wood is part of that. So it's not just a purely hearing related thing. So should we move on?
And we have the last question, Dr. Plack. What is the job market like for the hearing research trainee in academics and industry?
That's a really good question. I mean, I can only, when I say what's the job market like, I can only compare it really between different disciplines, I suppose. And my perception, at least, is that, and other people can sort of jump in and disagree with this, my perception is that when specialist jobs in hearing science are advertised, we don't have an enormous pool of applicants come forward. So I think there's to a certain extent, there's a dearth in qualified, PhDs and things like that. So my own perspective is that it's actually, in the UK, it's actually not that bad. I think that there is a bit more money going into hearing now. We've got biomedical research centres. We've got 3 of them with a hearing health theme, and so there's quite a lot of money going in. So I think there are opportunities in the UK for young researchers in different disciplines. I mean, if you wanted to join a psychology department, for example, there's a lot of competition with that. There's so many psychology PhDs, and so depending on what kind of job you want. So I think in terms of research positions, I'm not sure that I think the job market is reasonably good. I don't have any data on this. And I'm not sure what the situation is for industry. I don't really have any direct experience of this. Let's have a look. So we have another question, distribution.
Yes.
Yeah,
we
sorry.
Have one more question. The distribution of the noise exposure of people in music industry was not monopolar. Do you have any data to explain that?
Well, the actual graph I showed you was is two groups really. So it was a categorical distinction. I think it's between non-musical industry people and musical industry people. So it was, you know, it wasn't a random sampling of the population. So I, you know, sorry that might have been a little bit misleading, but there are going to be, you know, I mean, what I will say is that a lot of musicians now are clued up on hearing protection. We've got just up the road from the University of Manchester is the Royal Northern College of Music, and we've done some work with them. And a little bit frustratingly for us, but good news for the musicians. This is Sam Cooth, who's a lecturer at Manchester, who's done a lot of this work. A lot of these musicians now are wearing hearing protection, and they're completely on board with this. And so I think that is for music industry workers, that is going to be one of the big things. You know, are people prepared? Some musicians don't want to wear hearing protection. They think it diminishes their ability to hear their own instrument and the instrument of people they're playing with. But I think there's a recognition now, particularly with, you know, there's some really nice, I mean, I've got some myself, some musicians' earplugs that are sort of fitted. They're not cheap, but they give really good protection, and they have a really flat frequency response. It's a bit sort of late in the day for me, and my hearing's already been damaged through being in rock bands. But you know, so I think there will be actually, you know, in answer to the question, I suppose, is there is going to be, I imagine, a bimodal distribution, but that's rather to do with the people using hearing protection and people. Other people not using hearing protection, but it depends on the instrument you play. You know, obviously, if you're playing a very loud instrument, horn instrument, and you're practicing a lot in sort of a reverberant environment, then that might be a problem. So you know, I think it will depend to a certain extent what your role in the music industry is. But I think hearing protection is a big thing. You know, there are those who use hearing protection, and those that don't, and so that's likely to create quite a strong bimodal distribution now.
Well, Dr. Plack, thank you so much for your time, for your presentation. We're really glad with this session, and thank you so much.
You're most welcome. And again, thank you very much for inviting me. It was a different type of talk for me, but I very much enjoyed it. And thanks for joining me, and enjoy the rest of your days.
You too. So welcome to our series. We'll have another series maybe in August. So we will let you know. Thank you so much. Bye.